Hello everyone. So today we will be discussing metabolism, cellular respiration, and photosynthesis. So first of all, this is the way that you can see here on this picture that energy flows in our environment. So some energy is always released to the environment, to its surroundings as heat. But what takes energy from the sun are producers, and these are largely plants that are able to undergo the process of photosynthesis, which we will talk about today. Now, consumers are animals that eat or consume the plants, and that's how they harvest their energy. And that process is called cellular respiration, and we'll discuss that one in this lecture as well. Decomposers are organisms that essentially take energy from plants that have died or from animals. And so they decompose various nutrients. And so these are like fungi, bacteria, and worms that are able to undergo the decomposition process in the soil. And always some energy is lost to the surroundings as heat. So metabolic pathways exist, and we already learned a little bit about this in chemistry. So this will be a little bit of a recap. We need to discuss certain enzymes that function in cells firstly before we move on to cell respiration and photosynthesis. So there are a couple of metabolic pathways that happen in the cell. These are called anabolic and catabolic reactions. And anabolic reactions, if you think back to our macromolecules discussion, this is where you're taking small molecules or monomers, adding some energy, and then assembling them into larger ones called polymers. So like taking amino acids, putting them together into a large protein is an example of an anabolic reaction. Catabolic reactions are the opposite. This is where large molecules like polymers are broken down into small ones called monomers, and energy is released in this process. Now, what's interesting about these metabolic pathways is that they occur in the cell all of the time. We're constantly breaking bonds and building bonds. But what is interesting about them is that sometimes enzymes are used to help speed up this process. So enzymes, if you look at the next page, this is how enzymes work with their substrates. And substrates is just a fancy word for the reactants that are used in the process. So for example, if you're breaking apart a large protein, your large protein is your substrate or your reactant. And what gets released at the very end are your products. So these would be amino acids, for example. This is the way a substrate binds with its enzyme, and an enzyme is normally much larger than the substrate itself. So a little bit about this binding is that the enzyme has a certain region of it called an active site. And this active site is where the enzyme fits very specifically with its substrate. It's like a lock and a key. So enzymes are very particular in terms of their shape for their substrates. You can see it's not absolutely perfect, but it's going to create something called an induced fit, where they fit together very well and they sort of change shape a little bit as they fit together just to make the binding happen. So the binding happens on the enzyme. So the reaction is carried out on the enzyme, and then the bonds are broken for this large protein. This happens again on the enzyme. You can see that there's the bond that has been clipped in between these two parts of this protein, and then those two portions can be released as the products. So that's essentially how enzymes and substrates bind and work in the body. And we have lots of different enzymes in the body that help speed up reactions in this type of manner by binding to their substrate and then releasing the products. So how an enzyme actually works is through this type of enzyme-substrate model, creating an induced fit. Now, where the binding actually happens is the active site on the enzyme. That is the name of the site where the binding happens. If you take a look over here on the right-hand side of this picture, the reaction path is given to you on the x-axis, and the amount of energy is given on the y. And so this is looking at a type of energy called activation energy, which, as the name suggests, is the energy required for a reaction to proceed. So if you have your reactants and if you have your products, you can see that over time through the reaction path, as you follow a reaction without a catalyst in purple here, the reaction energy required goes up. And so you can think of activation energy as the barrier that you need to climb across in order for a reaction to proceed. So there's this barrier, and if you increase over that barrier, then the reaction will physically be able to proceed. Now, a catalyst is another way of saying enzyme. So this picture has the word catalyst, but it can mean enzyme. And so you can see that we need to get over quite a bit of an energy hurdle in order for the reaction to go forward. But what's interesting is where you have a reaction and you include a catalyst or an enzyme, that activation energy decreases. So you can see the dotted line in red indicates that the total activation energy, which was up here in purple, has decreased quite significantly here in red. So we only need to get over a slightly smaller hurdle in order for the reaction to proceed. And so because of this, activation energy is actually decreased when an enzyme is involved. And because of that, enzymes are able to speed up the reaction. So enzymes speed up reactions by binding with their substrates at their active site. They are able to decrease activation energy in order for this to occur, in order to speed up a reaction. Now, enzymes don't always need to be on in the cell. Sometimes they are inhibited. And so there are two types of enzyme inhibition that occur in the cell. So one's called competitive and the other one is called non-competitive inhibition. So competitive inhibition means that there is something in the cell that looks like the substrate. It's kind of what's called a substrate imposter, and it isn't necessarily exactly the same shape, but it's able to fit into the active site of the enzyme. And because it fits into the active site of the enzyme, it actually plugs the active site, so the proper substrate cannot come in and bind. Now, of course, there are several of this type of enzyme and several substrates. So if you think of a picture where you have lots and lots of substrate, lots and lots of enzyme, but now lots and lots of substrate imposters, they will plug up maybe half of those enzymes. So we still get substrate enzyme binding, but it's not quite as high as if there were no substrate imposter. So that substrate imposter is plugging the active site for some time. Now, a non-competitive inhibitor is a little bit different. It's also called an allosteric inhibitor. And what happens is the enzyme also has a secondary site on it called an allosteric region or an allosteric site. This can be shown on the next slide. Over here on the left, you have a picture of an enzyme with an active site, but it's also pointing to the allosteric region or the allosteric site. Now, what happens with an enzyme that has an allosteric site is that an inhibitor is made in the cell that comes into the allosteric site, binds to it, and that binding causes a conformational change or a shape change in the enzyme, where that active site is now closed. So if you use an inhibitor at an allosteric region of the enzyme to close the active site, that means your substrate, here in green, cannot bind at all. So this is a way of severely decreasing the rate of a reaction because now you don't get any binding. Whereas in the previous example of the competitive inhibitor, at least there are some enzymes out there that are not yet plugged and a substrate could bind quickly there. But here we have completely shut off the active site so that we get zero or very decreased binding. So that is the way of which an allosteric inhibition works. Now, if we want to reactivate that site, we can look over here on the right-hand side of this picture, and it shows allosteric activation. So the inhibitor leaves, and an activator comes in, binds to that site, because that active site is still closed. So an activator binds to the allosteric site. The enzyme is able to open back up again, and the substrate can bind. And some enzymes function in this way. Other enzymes function in competitive inhibition. So it really just depends on what the enzyme is and what the substrates are. As I mentioned to you, a non-competitive inhibitor will decrease the rate of a reaction severely versus a competitive inhibitor. So here in blue, we have non-competitive inhibition. Here in green, we have competitive inhibition, and a normal enzyme in purple. And so based on substrate concentration on the x-axis and the rate of a reaction, on the y, we can see the rate of a reaction severely declines for a non-competitive inhibitor because you are completely shutting down the active site. So moving on to cell respiration and photosynthesis, these are the equations that are used for cell respiration and photosynthesis. So just like the in metabolism that I just discussed, we have what's called reactants on the left-hand side of the equation, and on the right-hand side we have our products. So for cell respiration, our reactants are glucose and oxygen, and our products are CO2 or carbon dioxide, water, H2O, and ATP. And ATP is our energy source, also known as adenine triphosphate. Photosynthesis, on the other hand, you have your reactants listed here on the left-hand side: sunlight, CO2, and water. And on the right-hand side, you have oxygen and glucose. So these are your two important equations for the two metabolic processes that happen in the cell. Now, what you might notice is photosynthesis is largely a reverse of cell respiration. So if you know your equation for cell respiration, you just turn it around, where your products now become your reactants, and your reactants now become your products. Now that is except for sunlight. So plants are the only ones that actually use sunlight in the equation. So firstly, discussing aerobic cellular respiration. Aerobic meaning oxygen is used. So this happens in the mitochondria, and we've already learned the mitochondria is that little bean-shaped object that you can find in eukaryotic cells. And you have a whole bunch of folds, membrane folds here within the mitochondria, where the process of cell respiration actually occurs. And there's little enzymes in there that help speed up this process, and they're called ATP synthase enzymes. Now, I told you that the main source of energy in the cell is ATP. So here you see a picture of ATP, and what you can note is the base here, the ribose sugar, and then the 3 phosphate groups. And so you've already learned that phosphate groups are functional groups, and we have 3 phosphate groups, and so that hence the term ATP, triphosphate. Now, this is a renewable resource in the cell. One of the principles behind conservation of energy is you can never create or destroy energy, just convert it from one form to another. So whenever I say we make ATP, we're not actually generating it spontaneously. It's actually going from a form of a diphosphate to a triphosphate. So that 3rd phosphate group is either taken off if energy is used, or if we require energy in the form of ATP, so we're building it, we add the phosphate group back on. So that's physically what's happening in the cell. There are 3 stages of cell respiration: glycolysis, citric acid cycle, or Krebs cycle, and the electron transport chain. So I'll use the term citric acid cycle for number 2, but it can also be learned as the Krebs cycle in certain textbooks. So this is a picture of glycolysis, and within glycolysis there is an energy investment phase up here in these 5 steps, and there's an energy harvesting phase down here. Now you can see lots of structures starting with glucose, which is your beginning material for glycolysis. However, not just glucose is used in the body as a resource. So we don't just eat glucose, we eat lots of different sugars, we eat lots of fats and proteins, and so glucose is just used as an example in most biology textbooks as something that you start with and then you make energy from that. So we have glucose, and it's transformed into different structures as you're breaking it down. Essentially, you're cutting it in half, such that you have two structures at the very, very end that are called pyruvates. So this structure continues to get cut down into two of these pyruvates, but only one is shown here, which you can see at the end of step 10. So it takes 10 steps to get to this very end where we have two pyruvates. And you can see with the lines, you see underneath them a whole bunch of different enzymes. So hexokinase, fructose biphosphate adose. So there are lots of different enzymes involved in the process. And you can see that regeneration of energy is also outlined. ATP transferred into ADP. So we take a phosphate group off of it. Remember, that means we're using the energy. Whenever we take ADP down here, we add a phosphate group back on, we are generating ATP, so we are making energy. So glycolysis requires an investment of energy. You can't actually make energy in the body without investing some first. And so you invest 2 ATP molecules in this process, and you generate a total of 4, and so you have a net of 2 ATP. Now again, what you're making is pyruvate, and you have 2 of those molecules at the end, starting with 1 molecule of glucose. You also generate something called NADH. So you can see this NADH here. Now, again, it's a renewable resource, so NAD+ actually gets a hydrogen group added onto it, such that you generate this molecule called NADH. And NADH is an electron carrier. There are 2 types of electron carriers in the body for this process. They're called NADH and FADH2, which you'll see in the next step. Now, NADH, you can see this number 2 times. That means this cycle happens twice, or this sequence happens twice. And so we actually generate 2 NADH in the process of glycolysis. And essentially, you can think of these electron carriers as a bus. So you have this NADH bus, and you're strapping electrons onto it. And electrons are always released whenever you're breaking bonds. So you see lots of bonds being broken throughout this process. So you strap all these extra electrons onto this NADH bus, and essentially this bus drives all the way to step number 3 of cell respiration, called the electron transport chain. And at that point, all these electrons are dumped into the electron transport chain and used to generate the most energy. Because really from glycolysis, we only get 2 ATP, and that's a small number. After glycolysis, there is a very short mini step that happens just after. It's called oxidation of pyruvate. And so that pyruvate essentially needs to get transformed into something called acetyl-CoA, which is used in the second phase called the citric acid cycle. And so 3 steps, short steps happen here. Essentially, we're generating more NADH, 2 in fact, because we have 2 pyruvates, and we're generating some energy in the form of ATP as well. This is the citric acid cycle. So you see that input of acetyl-CoA, and the cycle happens once per acetyl-CoA. So for 1 molecule of glucose, you get 2 pyruvates, hence 2 acetyl-CoAs, so you get 2 spins of the citric acid cycle. And you can again see lots of different intermediary compounds that are generated from acetyl-CoA, and lots of enzymes, which are not written here, are involved in this process. And really the important thing to note is, again, you have that generation of NADH, which is your electron carriers, but you also see the release of a byproduct called CO2. You can see that down here as well. And you also see another electron carrier called FADH2. exactly the same function as NADH, we just have different numbers. So for one spin of the citric acid cycle, we actually generate 3 NADHs, 1 FADH2, 1 ATP, and lots of CO2. Now, all of the electron carriers that have been generated so far, which are NADHs and FADH2s, they all come in and they release their electrons into something called the electron transport chain. So you have the inner membrane here, the phospholipids you can see touching each other, hydrophilic head, hydrophobic tails towards each other. This is the membrane space. And you see a bunch of proteins or complexes embedded within it. So these complexes, what really happens is the electrons get bounced around from complex to complex until energy is generated. And so hydrogen ions are also released in this process. And what's important to note is that oxygen is used. So when you take oxygen and you take hydrogen ions, you generate water, or H2O. And so water is a byproduct that's released in the electron transport chain. Now, electrons are being bounced around from protein to protein, and eventually in the last protein called ATP synthase, that is where the majority of your ATP is going to result. And different textbooks will give you variations on the numbers, but in ours, we can essentially say that 28 or approximately 28 ATP are generated in this process. So you can see that's a large number compared to the previous two steps. If you remember back to how many NADHs we have, from one molecule of glucose, if you add up the NADH you generated in the previous two steps, as well as oxidation of pyruvate, don't forget that in between step, you actually have 10 NADH. And FADH2s, you only have 2 of those. So you're using the electrons from these 2 electron carriers in order to generate all that energy. And so I drew a picture of the overall process of cell respiration with the numbers. So glycolysis, step number 1, is here. This process happens in the cytoplasm. So something to note is that, yes, the majority of cell respiration happens in the mitochondria, but it begins in the cytoplasm. So glycolysis, you have 1 glucose, you break it down into 2 pyruvates, you invest 2 ATP, but you harvest or you make 4 ATP, so your net is 2 ATP from this process of glycolysis. You also generate 2 NADH electron carriers that will bring electrons to the electron transport chain. There is a mini step called oxidation of pyruvate that happens just after glycolysis, where again we generate 2 ATP and 2 NADH, and we convert those pyruvates to acetyl-CoAs. Over here, each of those acetyl-CoAs goes through its own citric acid cycle, where the output of each cycle are these numbers: 1 ATP, lots of CO2, 3 NADHs, and 1 FADH2. So in total, from 1 molecule of glucose, you would have to add up these numbers. So if we wanted total amounts for the citric acid cycle, it would be 2 ATP, 6 NADH, and 2 FADH2s. Now remember, the electron carriers go to the electron transport chain. So how many FADH do we have? We have 1 here, that gives you 2. How many NADH? We have 3 here, so we have 6. 2 more here, that's 8. 2 more here, that's 10. So we get a total input of 10 NADH, 2 FADH2s, and that's a total of 12 electron carriers in general, if you're not being specific. And so those electron carriers release their electrons through all these different complexes embedded in the membrane. Remember, we add oxygen at this process. That's why it's called aerobic cellular respiration. Hydrogen ions are released, and we generate some water in this process. And we produce approximately 28 ATP from this process of the electron transport chain for 1 molecule of glucose. So our total ATP production, taking into account 28 ATP plus if we go back 1 here and 1 here, that's 30, plus the net 2, that's 32. So anaerobic cellular respiration is a process by which we do not use any oxygen. And so that electron transport chain doesn't really happen in anaerobic cellular respiration. So you think about microorganisms that undergo fermentation to make alcohol. That's alcohol fermentation, and it doesn't use any oxygen. Or if you think about your muscles where you're exercising strenuously, and you're not allowing oxygen to get into your muscles, that's a buildup of something called lactic acid. And again, that's a type of anaerobic respiration. So how that works is, again, you have a molecule of glucose. You go through the process of glycolysis, which is step 1, and you generate 2 pyruvates. You have a net output of 2 ATP, but you do generate those NADH carriers as well. Now for the next part, you're undergoing a process called fermentation, and from those 2 pyruvates, you go straight to your end result, which is your lactate or your lactic acid. So from 2 pyruvates, you generate 2 lactates or 2 lactic acids. Same would be for alcohol, and you're using those NADH carriers. So from anaerobic cell respiration, you really only generate 2 ATP because out of those 3 steps from aerobic, you only use glycolysis and then a short fermentation step. Now, photosynthesis is essentially the opposite of cellular respiration, but I'm not going to go through any numbers that are involved in here. The process does not follow well when we try to assign numbers to it. So again, the overall equation is carbon dioxide plus water gives you sugar like glucose plus oxygen, and it happens in a region of the cell called a chloroplast. So this is a structure of a chloroplast, and you have these stacks of thylakoids within the chloroplast and lots of fluid, and really where the process occurs is in these little solar stacks. So these are the 2 steps of photosynthesis. On the left-hand side, it's called the light-dependent reactions, and on the right-hand side, it's called the Calvin cycle, also known as the light-independent reactions. By the name, you can hopefully understand that it's just the first process that relies directly on that energy from the sun. So essentially, we have sunlight, and sunlight is trapped in these stacks of thylakoids. And essentially a reaction occurs within it. Actually, several different short electron transport chains happen right in those little stacks. So we still have the electron transport chain, but it's actually embedded within the light-dependent reactions. So we have these little electron transport chains that happen right in these thylakoid stacks. We add some water in this process. You always need to water your plants. And we essentially generate oxygen. It's a byproduct for the plant, but it's really important for us. Something else that's generated is ATP because we are undergoing several steps of the electron transport chain within these light-dependent reactions, and we generate an electron carrier. And the electron carrier for this stage is called NADPH. Try to remember P in there for photosynthesis. So it's a little bit of a different carrier than in cell respiration. Again, there's no numbers involved. But these 2, ATP and NADPH, these 2 outputs from step 1 are used for step 2, the Calvin cycle. So the Calvin cycle relies on that ATP and NADPH to be used for cycling and generation of the product here, which is a precursor for glucose. So CO2 is required in this Calvin cycle, so that's the second stage of photosynthesis. And what is basically used from CO2 is the carbon. Carbon is used as a backbone to make the glucose because glucose is a series of carbons strung together. So the process of taking carbon from CO2 is called carbon fixation, and that happens in the Calvin cycle. So essentially, that's all of photosynthesis. You're using some of that carbon in stage 2 to generate your glucose. Again, there are no numbers in this process. The next couple of slides show a little bit more detail as to what's happening in the light-dependent reaction. So those stacks of thylakoids have these complexes embedded within them called photosystems. And these are little complexes that actually take those photons from light, trap them, and then electrons get excited and they are put through something called the electron transport chain, which you already know about. And so electron transport chains happen right in these photosystems. And so this is more of an overall picture on this next slide, where we have these photosystems here in green. You can see the electrons, the E's with the minus signs, being put through these various complexes. You can see energy from the sun here in these two yellow dashes. You can see hydrogen ions being pumped through as well. So we are using water in this case, breaking it down to oxygen and those hydrogen ions. We go through a couple of rounds of that electron transport chain. We are generating NADPH, and if we go down here, we are generating energy in the form of ATP. Those two are used in that second stage called the Calvin cycle, where we fix carbon from CO2, we use ATP and NADPH, and we make our sugar or our glucose. So again, this is a snapshot of the Calvin cycle if you wanted to see how the various intermediates look like. Again, we use carbon from CO2, we involve carbon fixation, and we generate that glucose. So cellular respiration and photosynthesis, those processes, because we have reactants that are products from one versus the other, it's actually a cycle within our environment. So plants use the CO2 that we output and they release oxygen for us and other consumers. And then animals or other consumers actually use that oxygen and release carbon dioxide. So we cycle within our ecosystem. And that's it on cellular respiration, photosynthesis, and metabolism involving enzymes. If you have any questions, please let me know.